Hey everyone, welcome back. Today we are going to finish up our water and sugar transport in plants section. I want to start us off today reviewing the pressure flow model that we went through at the end of the last video, and then move into some of the mechanisms for loading and unloading of phloem. So the pressure flow model, just to remind you, this is the way that sugars are moved in plants or translocation in plants. And it relies on the bulk flow of fluids through both the xylem, the water moving through the xylem, and sap or sucrose solutions through the phloem. And that moves by bulk flow. And while we learned that transpiration drives the movement of water through the xylem, what drives the movement of sap through the phloem is actually a Turgor pressure gradient. And so what I'm going to go through here in this pressure flow model is sort of reviewing how, going through the steps of how that Turgor pressure gradient is actually established. Just as a reminder, here we have a source leaf cell and we have an adjacent companion cell of the phloem, which is attached to that phloem sieve tube. And remember, in the veins of leaves, we've got the xylem running adjacent to those phloem elements. And so these are all, this is happening, this would be in a leaf, and then we've got a sink, and the sink shown here is a root cell. Okay, so the first step in this process it happens right here. And that first step in the process is to have sucrose actively moved from the source into the companion cell. So sucrose actively loaded is what the term we're going to use into companion cell from the source. This then establishes a high concentration of sucrose in that in the phloem. So what does that do then to the water potential in the phloem? If we have a high concentration of sucrose in the phloem, that's lots of solutes in the phloem, that solute potential then decreases. So our water potential decreases in that phloem cell. When the water potential decreases, then step 2 happens here. And so step 2 is shown right here. And that's water moving from that adjacent xylem into those phloem sieve tube elements. And so water moves with that water potential gradient, so along that water potential gradient, into adjacent sieve tubes. Great. So what's the result of that in our sieve tubes then? Well, as that water is moving in, with that water potential gradient, the result of that is that there's an increase in Turgor pressure in the sieve tube. So our Turgor pressure, our pressure potential, increases. So now we've built up pressure, and we've got this high Turgor pressure. We've got this high Turgor pressure in the sieve tube elements adjacent to that source cell. Okay, that Turgor pressure gradient, the Turgor pressure is high at the source, and then we need to lower the Turgor pressure to establish the Turgor pressure. We have to lower the Turgor pressure at the sink. And so our third step is actually going to happen right down here. And that is sucrose is unloaded from the phloem. And as we'll learn, this can either be active or, depending on what our sink is, this can either be active or passive. So sucrose unloaded from the phloem at the sink. So into a sink cell. And I'll go through where this is active and where this is passive, depending on the particular sink. So this is either passive or active. All right, so once we unload that phloem, what happens then? What's the result of unloading the phloem? And so the result of unloading that phloem, there's a couple of results. One is that our solute potential, the solute potential increases in those sieve tube elements. So solute potential increases in sieve tubes. Why is that? Well, as we're unloading the phloem, that means that there's less solutes in there, and so our solute potential ends up going up. And it increases in the sieve tubes, so the phloem part, until it's greater than that of the xylem. So greater than the solute potential in the xylem. And that's an important part, because as it's greater than the solute potential in the xylem, that's going to then cause water to flow back across out of the sieve tube elements, out of the phloem, and back into the xylem. And so it's that flowing across that then, so I'll write that down, H2O then moves as a result of increasing the solute potential by taking those solutes out of the phloem. Our water moves from the phloem to the xylem. Okay, what happens to the pressure potential then when the xylem, when the water moves from the phloem to the xylem? That pressure potential, the Turgor pressure, decreases. So Turgor pressure, write it out, Turgor pressure decreases in the phloem. And so that's happening right here. We have this decrease in Turgor pressure at the source. That decrease in Turgor pressure establishes, so I'll just put the 4 right here, it establishes this Turgor pressure gradient. And so step 4 in this is an established Turgor pressure gradient. Turgor pressure gradient in the phloem. And what's the result of that? Well, the result of that is that phloem sap is going to move by bulk flow. And so our phloem sap is actually going to follow that Turgor pressure gradient and move via bulk flow through the phloem. And so just a reminder that the transpiration and translocation are intimately related to each other. And this continuous loop of water moving through the xylem and back into the phloem, and through the xylem and back into the phloem, that continuous loop of water is actually necessary for that translocation to occur. So that's what allows us to drive this with a Turgor pressure gradient. All right, cool. That's complicated, but it's once we think about this in terms of water potentials, it's actually not too hard for us to grasp. So that's a cool result of moving sucrose in and out of cells at the source and at the sink. All right. So what's the evidence for the, there's a couple lines of evidence for the pressure flow model, and so I want to show you those. The first of these is one that we experience right now, and that is actually what's depicted in this picture here. And so what this is an aphid. Aphids are sap eating insects. And the reason I say that you're experiencing this right now is if you are out riding your bike on a nice sunny day and you're riding along the trail along Paradise Creek and you ride under a willow tree. Willows are especially known for this. They have lots of aphids in them. You're riding along that trail and you feel this light mist in your face and you think, oh, how refreshing that is. Well, you're gonna not think that's refreshing anymore after I tell you what that is. What that actually is this stuff coming out of the butt end of an aphid. And this is, we call it honeydew. And what honeydew is actually when an aphid inserts its mouth parts into the phloem of an actively growing tree. And so in the springtime, the sap starts moving in the spring. This aphid inserts its mouth part, and that pressure, that is the turgor pressure inside those phloem sieve tube elements, is actually so great that it pushes right through its body. And so this is the excess, this honeydew. And so you've got aphid shit flying on you. It's not actually digested, so I shouldn't say shit. But you have aphid, it's gone through the body of an aphid, misting you on the face. So that nice, gentle mist is actually not too refreshing if you think about what it is. But the reason I bring this up, it's funny, but the reason I bring it up is that this is actually good evidence for the pressure flow model. That pressure is what is forcing that out of the other end of the aphid, is the turgor pressure that's built up in those sieve tube elements. And scientists have used this for actually a long time as a way to measure the contents of the phloem sap. And so you can collect this honeydew and you can actually analyze it and measure contents of phloem sap through using aphids as your tool for extracting that phloem sap without doing much damage to the plant, since this is such a small proboscis that's actually being inserted here. Okay, so that's one line of evidence. I'll show you another line of evidence, but to show you the second line of evidence, we first have to understand how phloem is loaded and unloaded, and so how those sugars are actually moved into the phloem, because the second line of evidence depends on sort of manipulation of this process. And so when phloem is moved into and out of companion cells, which we call that loading and unloading. And so phloem loading is from source. So these are the photosynthesizing cells. These would be leaf cells from source to the companion cell of the phloem. So that's phloem loading, moving up from the source into the companion cell. Phloem unloading is from the companion cell, or the phloem companion cell, to a sink cell. And that sink might be a growing leaf, or that sink might be a root, some root tissue, or some other storage organ. And so those sinks are going to change, as we've talked about. This either happens, I mentioned, either passively or actively. And so this can happen passively if it goes with the concentration gradient. And so here we have depicted the outside of the cell with some molecules. This is just illustration of direct diffusion and facilitated diffusion. This is not specifically with sucrose. This is with any molecule that can move passively if the concentration outside the cell is greater than the concentration inside the cell, and the membrane can actually move. It can pass through the membrane. Membranes, remember, are semi-permeable. Some things can move through, some things can't. If they can't move through the membrane, then they can move through plasmodesmata by direct diffusion. And so this is along that concentration gradient. We've talked about this before. This is a passive way. If it can't go through the membrane, or if it can't go through plasma, or there's not plasmodesmata right there for it to go through, then we need to have facilitated diffusion. This is still passive. diffusion in general are both passive, but facilitated diffusion it moves these molecules via some transmembrane protein. And those transmembrane proteins just allow for molecules to move through with ease, so they don't have to go through that membrane. And so aquaporins, if you remember that, is a transmembrane protein for water. And this is a channel protein. Channel proteins are less specific than carrier proteins. A carrier protein usually involves some conformational change in the protein. And so these are more specific. And so these molecules drawn as these polygons here are specifically fit to this protein. And that protein, it fits into that protein and it changes the conformation and allows the molecule to move through. Sucrose actually needs to use more specific carrier proteins like this. But these are just ways that molecules can move from outside of a cell into a cell by diffusion, whether it's direct diffusion through the plasma membrane or through plasmodesmata, or facilitated diffusion through some channel or carrier protein, through a transmembrane protein. So what happens if we're actually against the concentration gradient? Well, if we're against the concentration gradient, then we need to actually do something different, and you actually have to use energy. And so against the concentration gradient these are active mechanisms. And just to remind you, remember, active mechanisms means that this actually requires ATP. It takes energy from the cell for this to happen. And so here we see a situation where we've got molecules like these red molecules here, let's call those sucrose, that are more concentrated inside the cell than outside of the cell. And we need to move those molecules from outside to inside. In this example, we have a molecule that's more concentrated outside the cell than it is in, but we want to move these out. And so we are trying to move against the concentration gradient. And so this is to move molecules against concentration gradient. And to do this, what a plant does, and our animals are no different than this, but what they do is they have active mechanisms that establish an electrochemical gradient first, and then use that electrochemical gradient to co-opt transmembrane proteins to move these against those, to move solutes against the concentration gradient. And so the way that electrochemical gradient is established is actually using something called a proton pump. And so here's a hydrogen proton pump, or if I label this as hydrogens, this is a hydrogen proton pump. We use ATP, using ATP to actually move hydrogen from inside to outside. This is just using where inside is a lower concentration than outside. So we've got more molecules on the outside of the cell than we do on the inside of the cell. This is moving against the concentration gradient, and so it takes ATP. So ATP is expended, and those protons move across the concentration gradient. And so the first step in this is use a proton pump to establish that electrochemical gradient. So now I have more protons on the outside of the cell than I have on the inside of the cell. And then I can actually use co-transporter proteins to piggyback on the concentration gradient that I've established with the proton pump. So I make that proton pump, and then there's a electrochemical gradient with higher concentration of protons on the outside than on the inside, and I can move those protons are going to move through by facilitated diffusion through these co-transporter proteins. But these proteins are special in that they're made to move both protons and a specific molecule in. And so that molecule can then move against the concentration gradient. So this can move against concentration gradient. If these move in the same direction as each other, if the protons and the molecule that we're really trying to move go in the same direction, we call this a symporter. If they go in opposite directions, it's called an antiporter. So in this case, I've got the molecule going out against its concentration gradient, and I've got the protons going in with the concentration gradient. And so what is the result of this proton pump establishing the electromagnetic gradient, this electrochemical gradient, is that we're actually able to move solutes against the concentration gradient by co-opting that, I'm just going to say, proton gradient, that hydrogen ion gradient, and co-transporters. So co-transporter proteins. So we're co-opting those co-transporter proteins to be able to move these things, like move these molecules against their concentration gradient. So symporters, same direction; antiporters, opposite directions. I'll just notice I'll just mention I used hydrogen as an example here. There are other ions, other ions that are used for this. There's things called potassium pumps, and potassium is another simple ion that's used to actually establish electrochemical gradients and move solutes in and out. An example of one of these was in the stomatal guard cells. They actually use potassium pumps to do this. So just you can establish electrochemical gradients without protons. Proton pumps are the most common, but those hydrogen proton pumps. But you also can do this with other simple ions as well. Okay, so those are co-transporter proteins, symporters and antiporters. So the reason that I bring this up is because this is how phloem is actually loaded at the sources. And so phloem uses a sucrose symporter to load the phloem. So remember, phloem loading was loading from the companion cell. So here we have outside of the companion cell and inside of the companion cell. And so this would be maybe at a source, like a growing or photosynthesizing leaf. And then this is loading that into the phloem. And what you'll see is that sucrose in the phloem, remember, is much more concentrated than it is outside of the phloem in that source cell. And so it has to actually move against that concentration gradient. And the way that this happens is that an electrochemical gradient, this proton gradient, is established by pumping actively using ATP, actively pumping protons outside and then establishing proton concentration. And we end up moving the sucrose in along with those protons through this proton sucrose symporter. So that's how phloem is loaded, through using a proton sucrose symporter. And so it's active mechanism. This actually takes energy to load the phloem. Okay, and so we can actually see more evidence of the pressure flow model and phloem loading and the importance of phloem loading through looking at mutants in this this sucrose symporter. And so there's the Sussman lab, Michael Sussman at University of Wisconsin, have identified, and this is a classic example now, they identified a sucrose symporter mutant. And that sucrose symporter mutant is shown here. Sorry, I'm talking and writing at the same time. That sucrose symporter mutant is shown here on the right-hand side, labeled suc2. And so that stands for sucrose. This is the sucrose mutant, and you can see that it doesn't do very well. The plants, compared to a wild type plant on the left, are actually quite small. They don't grow very well. And that is because they can't load very much sucrose into their phloem. And so if we take a look at the look at some anatomical evidence, if we look at the leaf, look at leaf cells, we have here, this is actually a vein. So this is where the phloem is. This has xylem and phloem in it. And we see chloroplast, and this is the wild type plant on the left-hand side here. And so we see some normal chloroplasts. You don't see very much here. There's not much to see, but we see these mesophyll cells that are photosynthesizing with their normal chloroplast, and then that vein adjacent to them. We see the same thing here in a suc2 mutant. And what you see is that the chloroplasts themselves are completely chock full of starch. And so the chloroplast, that sucrose is actually not able to be loaded into the phloem and be transported to the sink. And so it builds up at the source. These are source cells, these photosynthesizing leaf cells with their chloroplast, but now they're all clogged up with starch. And so and that's because this sucrose symporter, oops, that's not spelled correctly there. This sucrose symporter is not working correctly, and so you can actually then go ahead and measure how much sucrose is at the source and how much sucrose has made it to the sink. And if we look at this in both wild type and mutant cells, you actually see a pretty stark difference where at the, sorry about that, symporter, in a wild type plant at the sink, we have most of that sucrose is making it to the sink, to sink tissues, where in these suc2 mutants, we end up seeing that the sink tissues have a much less sucrose. And so we have a bunch of sucrose that's actually retained in the leaf. And so the way that this is done is you use radio-labeled carbon, and you can then watch where that carbon goes as it's made into sucrose and either moved into the sink or builds up at the source. Okay, so that's more evidence. That's evidence for the active transport for phloem loading. What happens at the sink then when we unload phloem? And so remember, sinks will change depending on what time of year we're looking at, what time of year this is. And so we have sinks sinks can be actively growing parts of the plant, like these growing leaves that we see right here. And so here's a developing leaf cell. That sucrose is being used for metabolism and biosynthesis, and so it's being eaten up as it's loaded into the phloem. And so the sucrose actually is a lower concentration in the sink cell than it is in the adjacent phloem. And so this can actually just go along the concentration gradient. So with our concentration gradient, and passively diffuse using a channel protein or a carrier protein. So using transmembrane proteins, it can move into the sink, that developing leaf cell, by passive movement along the concentration gradient. So that's pretty simple. That doesn't take energy. However, there are places where we're going to have to move against the concentration gradient, and a good example. And so unloading at sinks can vary. And so here's a sugar beet. As the name implies, sugar beets are full of sugar. So these cells in this storage, in this root storage cell, organ, these root cells are chock full of sucrose. And so they actually have more sucrose in them than they do. They have more sucrose in them than the adjacent companion cells do. And so what happens here is that to be able to, the way that these are, the way that this phloem is unloaded is actually by using what we call secondary active transport. And so that secondary active transport has to do with the fact that we're actually, this movement right here across the companion cell membranes and into the root cell is passive. It doesn't require ATP. It's against the it's with the concentration gradient. We have a relatively low concentration. Whoops, I shouldn't have circled that one. We have a relatively low concentration of sucrose in the cell itself, in the cytoplasm of that root cell. And the reason for that is that inside of that root cell, in the at the sink, you actually have the vacuole. And so this large vacuole here is full of sucrose. And so we have active movement via establishing a proton gradient and using that sucrose, using a proton sucrose antiporter. So these are going in the opposite direction. We use that proton sucrose antiporter to load these across the tonoplast. The tonoplast is just the name for the membrane of the vacuole. So these tonoplasts have in them these proton pumps, and they have the proton sucrose antiporters. And because of that, we can go against the concentration gradient from low concentration in the cytoplasm to high concentration inside of the vacuole that's now full of sucrose. And we can use that proton sucrose antiporter to move more sucrose into that while requiring ATP because we're establishing the electrochemical gradient using a proton pump. And so we call this secondary active transport across the tonoplast, because that requires that proton gradient for sucrose to be moved in. But this is the other way that the phloem is unloaded. So phloem unloaded into the root cells using secondary active transport. Okay, so that wraps up what we've learned about for translocation. And I want to now take a minute to sort of summarize this. There's a nice web summary that I have here, and I'll post this so you can look at it separately. I'll post this on our Blackboard site, but I think I should be able to show it to you here. It's just an HTML file. And I will go through this. This kind of summarizes both transpiration and translocation in one short animation. So I'm going to share a different screen with you to hopefully get this to work. Okay, so let's take a look at this here.
Plants transport water and dissolve minerals through a complex specialized tissue called xylem. Xylem cells form continuous columns from the roots through the stems and leaves. In mature xylem, the conducting cells are dead. Xylem also makes up the woody supporting tissue of trees, and one reason that trees can attain great heights is the vast amount of xylem they produce. Roots, stems, and leaves all contain xylem and play specific roles in the movement of water through the plant. Water and mineral transport begin with uptake from the soil. Water and dissolved minerals are taken up by the root hairs from the surrounding soil. Water moves through the root cortex to the vascular tissue. It passes into the xylem and moves upward through the root and into the stem. Mineral ions are pumped into the xylem by active transport. Water and dissolved minerals continue upward through the stem and into the leaves. Finally, the water reaches the veins in the leaf, where some is used to transport sugar in the phloem. The remaining water molecules pass out of the vascular tissue and into the mesophyll. In the mesophyll, some water molecules are used in the chemical processes of photosynthesis. Meanwhile, excess water molecules evaporate from the mesophyll cell surfaces into the moist interior of the leaf. When the stomata are open, the water molecules pass out of the leaf. This loss of water from the leaves through the stomata is called transpiration. A pair of guard cells controls the opening and closing of the stomata, which in turn control the rate of transpiration. Factors that influence the rate of transpiration include the availability of water in the soil, the relative humidity, the amount of sunlight, and the temperature. When there is abundant sunlight, low humidity, and adequate soil moisture, most of the stomata are open and the transpiration rate is high. On cloudy days with low soil moisture, most of the stomata are closed and the transpiration rate is low. At night, transpiration all but ceases. Plants transport sugars in a specialized tissue called phloem. Unlike the conducting cells in xylem, phloem cells are alive. Roots, stems, and leaves all contain phloem, and they play specific roles in the movement of sugar through the plant. The most commonly transported sugar in plants is sucrose. Sucrose transport begins in the leaves. The term source is used to describe an area of sugar production. Leaves, which are the main sites for photosynthesis and the production of sucrose, are the main sources in the plant. Sucrose is produced in the mesophyll cells. It is loaded by active transport into nearby companion cells of the phloem, and then diffuses through channels into the sieve tube members in the veins of the leaves. When the sugar concentration in the phloem rises near the source, water enters the phloem from the xylem by osmosis. This increases the Turgor pressure in the phloem and starts the flow of materials in the phloem. A sink is an area where sugars are used or stored. Roots and fruits are examples of sinks. While the direction of xylem transport is usually from roots to leaves, the direction of phloem transport is from leaves, sources, to sinks. In the area of the sink, sucrose is unloaded from the phloem by active transport. When the concentration of sucrose in the phloem near the sink is lowered, water moves out of the phloem by osmosis and back into the xylem. The difference in Turgor pressure between the source and the sink causes the flow of the solution in the phloem. Many factors influence the rate of sugar transport. Most important among these is the rate of photosynthesis. On sunny days, when the photosynthesis rate is high, phloem transport rates are also high. On cloudy days, when the photosynthesis rate is low, phloem transport rates are also low.
Okay. Wow. So that hopefully summarized a lot of what we've been talking about. Maybe I only needed to show you that video and you didn't need all of these other videos. Wow. I hope that's not true. In fact, you know, in listening through that, it connects a lot of pieces from the anatomy of plants and the anatomical parts, the tissues, the sort of form and function parts that we've been talking about, to the processes of water potential gradients and active and passive loading and unloading of the phloem. And so hopefully that little video connected a lot together. Like I said, I'll post that so you can watch that, you can study it, and you can use that. I'll post it separately, but of course it'll be at the end of this video too. Okay, I'm going to end there, and we'll see you next time.